My name is Bill Noble. I'm from the University of Washington in Seattle. When they asked me what I was going to talk about, I came up with a very broad title because I didn't know what I was going to talk about. So what I'm actually going to talk about is methods for imputing missing data. And we'll go through two different projects that different graduate students in my lab have done. One of them that's just about to appear in Nature Communications and one that's still underway. And using different kinds of machine learning methods. So actually Yunjun's talk was nice because it introduced at least some of the concepts that I'll be drawing on in this talk. So we've got 45 minutes total. I'm happy to entertain questions throughout. And then I'll just figure out how to make it fit in the allotted time. As Yunjun also provided a nice backdrop because a lot of the data that I'm going to be talking about is drawn from the Encode consortium, which she also alluded to, and from the Roadmap Epigenomics Consortium. Encode stands for the Encyclopedia of DNA Elements. It's a follow-on to the Human Genome Project. It was started in 2003. And the idea is, the goal is to build a catalog of all the functional elements in the human genome. And so what we've been, my group has been involved in Encode since the beginning, and we're basically trying to develop methods in that context to try to make sense of the large amounts of data that Encode and other kinds of consortia like it are producing. So Yunjun alluded to some of the kinds of data, but let me just go over what types of data we're looking at. In Encode, basically all of the data is high throughput sequencing data, and it measures different aspects of the human genome. So this is a figure from one of the overview papers that Encode published back in 2011. Some of the data is going to be about how genes get transcribed. So the transcription of the genes into messenger RNAs, so that's like RNA-seq data, for example. Some of the data has to do with protein binding. There's two different classes of protein binding events. Some of them are transcription factors that typically bind and punctate regions of the genome and are implicated in gene regulation activity. There are also histone modifications like Yunjun talked about, where the histones are the guys inside of these nucleosomes here, and the tails of those histones have modifications along them that are implicated in chromatin architecture, the structure of the DNA in the cell. And also sometimes in recruiting the transcription factors that do the gene regulation. And then there's also some kinds of measurements that just have to do with sort of local packing properties of the DNA. The one that we use most in this analysis is called DNA sensitivity, although there are other assays like ATAC-seq that measure similar kinds of things. Okay, and then also an additional level of complexity in this data is that in addition to having many different kinds of assays, different types of experimental assays that you can perform, they can be carried out in lots of different cell types or cell lines and so on. So this is just a figure from the Roadmap Epigenomics Consortium just showing schematically the different types of tissues from which data was derived. But conceptually, when we think about this data, because it's all high-throughput sequencing data, you can think of it essentially as each experiment yields a vector, where the length of the vector is the length of the human genome, and the values in the vector represent sort of the experimental signal at that position. Often we don't work at base pair resolution. In most of this work, we'll be working at 25 base pair resolution, so you sort of average in a window of 25 bases to get the signal value. Nominally, all of these assays measure essentially something related to the depth of coverage, so how many short-read sequences overlapped with a given genomic position. The differences in the assays have to do with how the short-read sequencing is prepared. So you're enriching for different kinds of phenomena, and that allows you to see different aspects of genome function. So in this picture, you've got 3 tracks. Each track corresponds to one particular assay. So these are the names of the cell types here and the assays below it. So HSMM is one cell type. This is an adipocyte cell type, and so on. These are the names of 3 different kinds of histone modifications. It doesn't really matter for this talk, but they'll just to tell you, these weird acronyms stand for things like this is the 3rd histone, the lysine at position 27 along the tail, and it has 3 methylations on it. So there's a lot of different kinds of histone modifications that you can measure for any given cell type. And you can also measure lots of different transcription factors. So the result of all this is that you get a lot of data, basically. Here's just a subset of the data so you can see these are different kinds of histone modifications listed along here, plus one that's the DNA, that's the chromatin accessibility one. And these are different cell types. And the colors represent whether or not that particular experiment has been done yet. So some cell types, these are called the tier 1 cell types. These are the ones that lots of the labs within Encode have focused on. And so many, many different assays have been performed in these cell types. There are also some assays that are popular. People think they're more informative or whatever, and they end up getting done in many different cell types. But of course, you still have lots of other experiments that haven't been performed yet. So in general, it's a fairly big data set. I think this is somewhat old, but as of some months ago, there were about 5,700 experiments that had been performed within the Encode portal, so you can go and download all of this data if you want to. Each of these experiments covers the whole 3 billion base pairs of the genome, but the matrix itself is only about 5% complete. This is actually a picture of a subset of it. It's less complete than that. And so the task that we're interested in is essentially to say, well, maybe we haven't done that experiment yet, but we would really be interested in knowing what what's the result of that assay in that cell type. Obviously, you can go do that, but a high-throughput sequencing experiment is hundreds of dollars, typically, something like that, and maybe that's an expensive proposition, so maybe we can try to predict it. So we think of that as an imputation task, and we see whether or not we can predict this sort of systematically for all of these different experiments. So this is a task that was first done by Jason. Is Jason here? Oh, well, he's not here, but Jason and Manolis Kellis published a paper solving this problem in 2015 called Chrome Impute. So this was at the same time that a graduate student in my lab, Tim Durham, was developing what turns out to be a very different approach to the problem. So we're going to talk about what Tim did, and then compare and contrast, and then we'll talk about what a subsequent graduate student is currently doing to solve the same problem with different methods. So the people involved in the project, Tim is the main grad student, Max is another grad student or former grad student in my lab who helped out with the project. Jeff Haubert was a research scientist, now retired, who actually specializes in mass spectrometry, but I'll tell you about his role in a second. And then Jeff Bilms is a longtime collaborator of mine. He's a machine learning expert in the electrical engineering department. So one way that this project started was discussions with Jeff Haubert. He had been involved, one of the ways that he switched from becoming an experimental, he's trained as an analytical chemist, and he switched over to doing machine learning late in his career. And the way that he learned to do machine learning was in the context of the Netflix challenge. So he thought machine learning was cool. He heard about this problem, which was that Netflix said, we've got this huge data matrix. We've got lots of users of Netflix. They've told us some of their movie preferences. Can you impute the remainder of their movie preferences? Jeff ended up being on the team that actually crossed the finish line first for the Netflix challenge. Netflix had a held-out validation set that then anybody who crossed the finish line within something like 24 hours after the first team crossed, they would then test it on the held-out set. And on that set, his team was second, so he didn't actually get the prize, but he learned a lot in the process. So let me just tell you about the kinds of methods that ended up being used in the Netflix challenge. The basic idea, and Yunjun alluded to these, is to decompose the matrix that we're interested in into two different smaller matrices. So you have some latent space, a k-dimensional space that you're using to represent the preferences of users and also the features of movies. And the key idea is something like this. Here's Tim. Here's a movie called Breaking Away. And you want to be able to predict whether or not Tim's going to like that movie. So you have one row in here that corresponds to the factors, the latent factors that correspond to this movie. And one of those might be, how much is this movie about cycling? And then you have one column here. One of those is about Tim, and this is how much does Tim enjoy in movies about cycling? And the answer is he likes them a lot. And so probably it's going to predict that he has a strong preference for this particular movie. So the question is, could we just apply this kind of technique more or less out of the box to the imputation problem with the Encode data set? And the only real challenge there is that the data is a little bit different in the sense that it's a tensor instead of a matrix. So it's three-dimensional rather than two-dimensional. You have the assay types here, the cell types here, and then millions of positions along the genome because we're working at 25 base pair resolution. But nonetheless, you can still frame this as essentially a tensor decomposition task, where instead of two matrices, you have three matrices. Here we have L factors that correspond to each cell type, L factors that correspond to each assay type, and L factors for each position along the genome. And then when you want to do an imputation, you can basically just take those factors, do the scalar products of them, and add them up to get you a predicted value. Okay, so this is, oh yeah, question.
Are you keeping track of the DNA sequences at all, or just?
No DNA sequence. So in principle, you could, right? You could add an additional column or multiple columns that told you maybe features of the sequence, but you would have to fit into this framework, you'd have to have some kind of featureization of the DNA to kit it. You could do Kamer counts or something like that. There was another question. No, maybe not. So this generalization to 3D is also not novel. It's been done before. There were actually two groups. This is one of them that did it in the same time back in 1970. It's called ParaFac, and it's essentially just exactly what you'd expect, where you generalize this from a 2D to a 3D setting. One thing that we added in here, so let me just show you this. This is the optimization, the loss function that we're optimizing. What you have here is essentially the sum of these scalar products here. And you're basically saying, I'm going to look at the mean squared error between the training set values that I'm holding out. So you're doing imputation on some subset of values where you know the answer, but you don't tell the system. And you look at the prediction on the right-hand side and the actual training value there and try to minimize the mean squared error between them. A key component is that you also have some regularizers over here where you're trying to keep the L2 norm of each of these matrices relatively small. So there's these hyperparameters, lambda, A, C, and G here that control the strength of the regularizer. Yeah?
Can you just walk through how the dimensions of that matrix multiplication work out? Because it seems like when you multiply A with G, you end up with something with 24 rows, and then that doesn't work out with C multiplying.
Can I? Maybe I did it. So each of these has an L, okay, right? So you have a JL, a KL, and an IL right in there. All right. It may be the picture's not very. All right. Okay. Okay? So that's the basic idea. So for the purposes of developing the model, we used a sort of a better a supported subset of the data. We had a requirement that each row and each column have at least 5 examples in it. So that rather than having like 95% missing data, we ended up with like 33% missing data. Yeah, so 1,014 data sets were available out of 3,048. And then we split this into a training set, a validation set that we use for selecting things like the regularization parameters, and then a test set of 20%. So one of the challenges here is that it is pretty big data. And it turns out that training these things is kind of difficult to do just from a purely computational perspective. So one of the things that Tim had to learn how to do was do all of this in the cloud. And so we did this using a parallelized stochastic gradient descent. Essentially, you're just computing the gradients at each step, but you do it in small batches. And the way that it works is you basically farm out subsets of the data. You have some master node here, and it's farming out subsets of the data. And they're each doing some rounds of stochastic gradient descent, updating their parameters. And then, so it does 5,000 iterations of the local parameters for one subset of the genome. And then it updates, so it averages the assay and cell type parameters from all the different parts of the genome together after those 5,000 iterations, and then farms those back out and does another round, and so on. After you've done the whole thing, you can actually then go through and compute the genotype parameters if you fix the cell type and assay type parameters, then you can deterministically figure out what the appropriate genotype parameters are, because that's a convex optimization. Okay, so I think this is sometimes interesting for people to hear. This was an exciting adventure in learning to use cloud computing. This is just a little timeline to show you how that went. As you can see, we were switching from different systems. We started in Hadoop, we switched to Spark, and then we switched over different variants of Spark. Sometimes these were driven by, at one point there was a bug in one of these systems and we had to switch to a different system because they said that won't get fixed till the next release. But mostly it was driven by where we could get money. So, we got a poster award for 10 grand and then another award of 20 grand, but then this was a bug on Azure. And so while we were waiting for them to fix that, we got an AWS award and switched over there. And eventually we ran out from AWS, switched back to Azure, and then got another award and switched to So one lesson is you can blow through a lot of cloud dollars fairly quickly as you're trying things out and experimenting with different kinds of techniques. The other lesson is that if you have at least a compelling story, you can usually get those dollars without actually paying any of them. So the cost to us was zero, but the cost on paper was a lot higher. Yeah.
When they were developing it, was he developing it at scale, or it just sort of surprised me that you're blowing through that much money for that length. I mean, it seems like you could take a smaller data set, or maybe you had the money.
Well, for one thing is, remember, it wasn't actually money, right? It was all just on paper. So we developed, there were some implementation testing, to make sure that it ran on smaller versions of the data. But a lot of the testing involves choosing regularization parameters and making decisions about whether or not this approach is going to work well. And realistically, choices made based on small data about things like model architecture just don't generalize. So you have to test them at scale, unfortunately. There was some other? Okay. So the nice thing is that, just qualitatively, here's a picture just showing pairs of tracks. So this is the imputed data on the top, and this is the observed data from the held out test set, right? So you can look at it and you can just see, okay, it does seem to be doing qualitatively a pretty good job. Each of these pairs, they look pretty similar. And this is not cherry picked. This is just more or less what it typically looks like looking across the genome. The other thing is that you can go in and look at the latent factors learned by the model and see at least some interesting patterns that look like it's picking up on something. So what we're doing here, there's 100, so I didn't mention this, one of the key hyperparameters, of course, is that L, right, the dimensionality of the latent space. And particularly with the multiple rounds of review, we had to do many, many expensive experiments to try different variants of L. And it turned out that 100 worked pretty well. You got down to minimum, I think the next best performance was at something like 68 parameters. We tried, I'm not going to show you all the plots, but basically 100 was the minimum, and then it didn't get any better when you went up to hundreds and hundreds of parameters. So these are the 100 dimensions here. And all we've done here is sort of make a meta gene. We've taken the GenCode gene annotations and sort of averaged all of these 100 latent factors across all the genes in the genome, and then plotted them in this heat map here, which is kind of hard to see in this light. But the key point is that there seem to be some of the factors that are picking up on different features of the gene. So you can see, for example, changes in, there's an enrichment along here for things that are enriched in the 5' UTR, and so on. And there's other parts of it that are picking up on different gene components. We also looked at things like data that wasn't even used in our assay. So P300 is a particular kind of mark that is known to mark enhancers, often marks enhancers. And these are just patterns of the 100 latent factors over lots of different P300 sites, so averaged over many different P300 sites. We've just segregated the 100 factors based on whether they have a peak in the middle of the P300 site or they have a trough in the middle. So there's, I think, 59 here and 41 here, but you can see there's clearly a pattern there. It's noticing something's going on. And then we also spent some time making sure that the method could work well when you have relatively few data sets for a given cell type. Because one of the key applications here would be, here's a cell type that hasn't really been studied very much. We only have 1 or 2 different experiments performed. And can we successfully do inference of other types of experiments in a cell type when you really don't have much data? And so this was basically doing simulations where we would toss out some of the cell types, some of the data that was available for a cell type, and then check how the model performance degraded. This is if you, this top one, the orange one here, is if you train only on, you're using all of the data except you're enforcing that in this one cell type, we only have a single assay, and it's this one histone mark H3K4ME1. The green one is similar except using a different mark. And it turns out you can see consistently the orange seems to be worse in the sense that it's a higher mean squared error for all of the ones where we did both of those, which is consistent with what people know in the field, that generally speaking, the green mark, this ME3, is thought to be a more informative kind of histone mark. And then in general, if you train on the full data set, which is the 5-fold cross-validated blue one here, especially comparing to the green, you don't see a huge degradation in performance. And so this was encouraging that it seems to work well when you don't have many data sets available. We did, of course, do systematic comparison to other methods. Basically, the only other method is the one I mentioned, Jason Ernst, Chrome impute technique. We also compared to a sort of a baseline model called the main effects, which I'll explain on the next slide. For the purposes of showing the comparison, we use these what are called ternary plots, which I had not heard of before, but Tim found somewhere. They're kind of cool. Basically, it's this projection where you can compare 3 things. You're basically taking the mean squared errors or whatever your performance measure is, and projecting into a space in such a way that the sort of section of the space in which you reside shows which of the 3 methods does best. So we're color coding them according to, like, the pink area here. Points over there would be ones where predicted was the best, and similarly for the others. The main effects model, I'm just going to mention briefly, is a model that we included as a baseline because it actually works pretty well in a lot of recommender systems. And it's basically saying, if we're going to predict what's the particular value for this cell type in this assay at this position, we're first going to use the global mean value for the entire matrix. We're going to subtract that out, and then we're going to look at the residual and take the average for that cell type, and then we subtract out those cell type residuals, and you take the average for each assay type, and those 3 values added together give you an imputed value for the given position. And that turns out to be a really very simple model that actually, for a long time, was hard to beat, honestly. It works pretty well. And so what we see is that compared to and I didn't mention what Chrome impute does. So Chrome impute is an ensemble of regression trees, and so it's actually training a different model for every possible imputed experiment. So it's essentially selecting training examples from the row and column of that matrix that I showed you and saying, to predict this missing value, I'm going to look for other assays in this cell type and other cell types with this assay and build up a set of features and train a model for that one. So it's a very different style of model. What we see is that if we use the mean squared error, so the global mean squared error between the real and the predicted data, the predicted is outperforming Chrome impute. So this is just that plot, and I've written down what percentage of the dots are within each of these 3. They don't always add up to 1 because some of them are directly on the line. Sometimes they tie. But 62% of the time, we're getting a better mean squared error from predicted. On the other hand, yeah, question.
What's a dot?
Oh, sorry. Good question. Each dot is one of these assays, or say one of these experimental tracks. So it's a combination of a cell type and an assay type averaged across the entire human genome.
How many dots?
So this is in the test set. I can't remember, the number was it's on the order of 200, I think, something like that. And so oh no, wait, this is cross-validated, so it should be, is it 1,000? That would be 1,014 if it's, I think this is the full cross-validated result. And the key thing is that on the mean squared error, we're doing the best, but this is, in a sense, not that surprising, right? Because the mean squared error is precisely the objective that we're optimizing for.
Did you retrain Chrome Impute?
Yes, right, so that's a good question. And we did not retrain Chrome Impute. So there is an unfairness here, because Chrome Impute is trained in a leave one out setting, right? So we're doing a cross-validated 5-fold cross-validation, and they're doing a leave one out cross-validation. So it wasn't possible for us to retrain Chrome Impute, and it's not really feasible to do leave one out cross-validation with this model. So that does give them something of an advantage. If you switch to looking at something like genome-wide Pearson correlation, then the Chrome Impute is still doing better than predicted on that performance measure. And then there are a number of other performance measures. Here's one that I think is interesting. This is if you take the top 1% of the observed data, so you're kind of looking at the peaks, and then you ask what percentage of the signal value is found in the top 5% of the imputed windows, right? So you're sort of asking how well do the true peaks in the data get called as peaks in your imputed data. And again, there, Chrome Impute is doing better in that setting. In general, we'll see that Chrome Impute does a really good job of identifying and modeling peaks, although we also see that it tends to, on average, have an over-prediction, so it tends to predict higher values on average.
Okay.
Yeah.
Is any of this chromosome specific, or
Is it what specific?
chromosome specific?
Oh, good question. So the question was whether this is chromosome specific. We basically see similar trends. I don't think I have that plot in here. I do have a plot like that for the next method, and you see similar trends across all the chromosomes.
So is this kind of implying that the data has a particular sparseness structure that feature selection actually helps with prediction? That's kind of what's being implied here.
Well, I don't know, because there's so many different possible explanations. So that's certainly one plausible one. Yeah.
Yeah, I mean, on that similar note, I was thinking actually one of the explanations would be that this is a linear model, right?
Mm-hmm. Yeah, that's wait for part 2.
Okay.
Yeah.
What are your sentiments on using Pearson versus Spearman? Is Pearson's like sensitive to outliers?
So we did ArcSine transform all of these data to deal with outliers first. So everything is, I should have mentioned that before, all of this is ArcSine transformed. But we basically, these performance measures, we were trying to be consistent with the Chrome Impute paper. So these are the measures that they used. Wow, we got lots of them, yeah.
Is there anything peculiar or interesting about the cases where the main effect model is?
Yeah, so we haven't done a lot of looking into that. I mean, I think it is something that we should investigate. I suspect that this is, cases where we really aren't regularizing enough, right? I mean, if you have this 3-parameter, or it's not 3, but 3 per position model, it implies that some kind of regularization might help. But so far in our hyperparameter search, we haven't found where that sweet spot is. Yeah.
Yeah, did you try an L1 norm?
So we haven't tried an L1 norm, but it's an obvious thing to try. Yeah.
Sorry, one last thing was, have you looked at like a mutual exclusivity metric? So what I often found is Chrome Impute makes mistakes which are biologically very obvious. So it overall does very well, but in situations where a gene is expressed, the promoter has to be active. And it will often make a mistake.
Oh, I see.
In situations where you see HTK36ME3 on the gene body, which
Mm-hmm.
means the gene is expressed, the promoter looks like it's bivalent, right? So it will have K4ME3 and
Right.
K4ME3. I think part of the reason is it can't look that far, and also it's not exclusively. So I think those are biological situations that are simply implausible, and those mistakes I would consider most egregious. So I think it would be interesting to see if some of these other methods actually, are able to go around it or not.
Yeah, although I wouldn't expect, there's nothing in our model that captures the genomic axis really either. So it would be an interesting measure, but a priori, I don't necessarily think we might fix that problem. Yeah. I don't know the answer to that. So the question was about cell type specific marks. So you mean, so actually what do you mean by a cell type specific mark?
Are specifics in just one cell types and not the
Oh, I see. Good question. I think that I don't have any numbers for that, but I can find out from Tim, I think is the short answer. Yeah.
Does it change also from different types of marks or DNAs versus?
So there's a yes, but it's not an obvious like, oh, one model's better at this class. You look at which ones it does better at, and there's not a clear trend. So this is just the same data represented a different way. I just want to point out one thing. So these are just showing the log fold change for each of the 3 performance measures I showed you on the previous slide when you're comparing pairs of methods. So for example, we know predicted is better than main effects. Mean squared error smaller is better. So the negative log fold change is good and positive is good here. And so this is again showing that predicted is better than Chrome Impute and mean squared error, but worse in these 3. The thing I want to point out, though, is that if you look at the correlation, before I point that out, Sushmita, yes?
You can finish that.
Okay, if you look at the correlation between the 2 of them, it's actually quite low. That is, the predicted and Chrome imputed values don't actually agree with each other, which is not that surprising because they're like radically different kinds of predictive models. And so the obvious thing to try was, well, what happens if we just average the 2? So you make a prediction where now you get the Chrome impute prediction and the predicted prediction, and you sum them and divide by 2, and that actually does better again. So we're now 73% of the time better in terms of mean squared error. We're now doing better in terms of the correlation as well, although it's still the case that Chrome Impute really likes to do well on the high peaks. So now, Sushmita.
So I was wondering, so I guess because you are putting all of these different marks together, you are able to also incorporate because there's correlation between the marks, and the marks are kind of mutually informative.
Yes.
But I was curious what would happen if you were to take one data type at a time and then see how well that performs, or somehow maybe your main effect model is capturing that. So you do an imputation using, say, just FDNA is 1. You do imputation using just K for ME3. How does that compare to this joint?
I mean, we kind of did that in the plot I showed before where we were restricting it to using cell types where we just had H3K4ME1 or just H3K4ME3. But we didn't do it systematically for every assay type, partly because it's not that common to have precisely one assay done in a given cell type. Okay, so let me now go on to the next one because there's not that much time left. So enter second graduate student, Jacob Schreiber. Jacob now developed a piece of software that follows on predicted. He has a theme. All his software is named after fruit. And so the fruit that he chose was an avocado, and this is supposed to be an imputed avocado. It's got some missing. So that's what you're going to see on all the slides. Tim is still involved, but Max Liebrecht left for a faculty position, and Jeff Halbert retired. So they're not involved in this part of it, unfortunately. So there's 2 main differences between predicted and avocado. One of them is this. So we're going to try to add some nonlinearity. The idea is there's an existing class of methods called deep tensor factorization. So this is sort of conceptually what predicted is doing. You're basically taking your matrices and you're performing sort of a scalar product on all of them and then putting that in as your prediction. In the deep tensor factorization, you still do an element-wise combination of these factors, these 3 different kinds of factors, but instead of then summing them and calling that your prediction, you put that into a deep neural network. And so then you can incorporate nonlinearities. Jacob thought, well, why do we have to actually do this element-wise combination? Why not stretch out these 3 factors side by side? Now there's no longer the requirement that they have the same dimensionality. You can have different values of L, an L sub C for cell type, an assay and position, and so on. And you still get the benefit of doing the dense layers. And so that's one difference. The other difference is that in thinking about the model itself, the vast majority of the parameters of this model are devoted to modeling the locations along the genomic axis. And so the question was, could we do something better than just having one big matrix that has, about 1.2 million positions just in the Encode pilot regions, just in the 1% of the genome, and 100 position-specific factors for each one. And the idea is that if we think about what's going on biologically, we know that there are different sort of scales of phenomena that we're interested in. There may be some punctate phenomena that we really care about, sort of 25 base pair resolution things, but a lot of the things we're interested in are sort of the scale of a nucleosome. So maybe we should be looking at averaging and having factors that look at 250 base pair resolution, or even like 5,000 base pair resolution for domain scale activity. And so the other difference between Avocado and Predicted is that Avocado has some factors that are done at 25 base pair, some at 250 base pair, and some at 5 kilobase pairs. So as Yunjun alluded to, you can now, there's lots of different things you can try. You now have structural parameters for the Avocado model. You have to choose the number of layers. You have to choose how many neurons within each layer. You have to choose the number of factors for cell types, for assay types, and then for each of these 3 different resolutions of the position. And this doesn't even mention all of the other sort of implicit hyperparameters about what specific kinds of regularization strategies you're going to use and so on.
Can you clarify the size of the input for each of those 3, the cell type, assay, position?
The size of the input? What do you mean? So the,
I think it's like 1 hot encoded, and the embedding is learned in the weights connected to a particular model encoding. Yeah, it's like word embedding.
yes, that's what she said. So what Jacob did for this part was essentially just do random search. You define a grid over your hyperparameter space. So you have a 7-dimensional space with a sort of plausible range of values, where plausible is defined a priori based on intuition. And then randomly choosing points within that grid. And we did 1,000 of them, and this is just on a validation set. So we have a 764 training set and a validation set of 100 tracks. And we're looking at the mean squared error as a way to select the parameters. And so the best one here has a MSE of 0.1189, which at least on this validation set, which obviously we've already selected on it, but it's still looking good in the sense that we're getting an improvement over both the MSE from Predicted and from Chrome Impute on these same tracks. If you look in more detail at what's going on in that random search, it seems that the main factors are sort of the number of neurons per layer. So this is a plot showing the distribution of mean squared errors for all 1,000 models as a function of the number of neurons. So we have 128 up to 2,048. And it seems to imply that it might even be a good idea to go to even larger ones, although then it starts to get even more expensive. We're running out of GPUs in the lab. And the neural net layers also seem to be helpful. Yeah. So if you try Predicted with these genomic factors that span longer regions?
No.
Break down how much of this advantage is coming from the non-linearity versus that?
Right. No, we haven't done that. Partly because like these results are only a couple days old. But I agree that would be because we have these 2 big changes, and we should definitely tease that out. I think Jacob did, because he came up with the idea of subdividing later, he did do Avocado without that, and it wasn't working as well. But of course, we've now tweaked things since then, so we should return to do that. We didn't. Oh yeah, question.
Sorry. Is the region size one of these random parameters, or is it something that you're?
No, the region sizes we fixed. So the 25, 250, and 5 KB, that was just, we said, we have some intuitions, we're just going to fix that. I mean, you could vary that as well. Yeah.
Is there anything going on with that thing looking bimodal?
Oh, good question. So I think the bimodality here has to do with the layer, how many layers you're looking at. At least that's my guess, but I haven't dived into that myself.
Is the picture accurate? Did you have 2 layers in the or how many?
No, that was just a conceptual picture. So we end up using, I think we end up using 4 layers for the guy that's at the bottom. So, again, we define this grid a priori. It looks like we're at the edge of the grid in 2 of the dimensions, so we might want to go even larger.
Yeah, it seems like you have enough data to run deeper models.
Yes, I think that's right. It's mostly about compute resource, yeah. So if anybody has extra GPUs they don't want, we can make use of them. We didn't do that much playing around with different regularization strategies, but one that Jacob tried is Dropout. This is a regularization technique where you basically skip over updating some of the weights in some of the iterations, and it seems clear, so there's a rate, so how often you drop, or what percentage of the weights get dropped out at each round, and it seems clear that doing dropout is better than not doing dropout, and the particular value of the dropout doesn't make that much difference, but it does give you a little difference. This is the difference here between without dropout and with dropout. So, yeah. Question? Yep, you. So, sorry, these are each different values of dropout, and each dot is one of the Avocado models, and then the line is the best that we got from Avocado here. This is the lowest value, and then the other lines are just for reference. Those are the lines for the other models. Sorry.
So,
yeah.
very basic, you may have said, what is dropout?
Dropout is a regularization strategy in which you, during training, you skip over updating some of the weights at each iteration. So it's a way to reduce the chance of overfitting. Okay, so I'm supposed to be done in 3 minutes, so let's see, I want to just mention, so there's a number of different performance measures that we go through. So this is just showing that when we actually skip from the validation set to cross-validation on the Encode pilot regions, that we maintain good performance in terms of the mean squared error. You can see that we're outperforming, so we're looking at, there's two different complementary measures here. This is the percentage of the top 1% of observed data that's found in the top 5% of the imputed data, and then this is the converse of that, where you take the top 1% of the imputed data and see if it's found in the top 5% of the signal of the actual real data. And what you can see is that in terms of this first measure, Chrome Impute is still doing better, but on the other hand, Avocado and Predicted are doing better than Chrome Impute on the other measure. So that's sort of consistent with this idea that maybe we're getting over-prediction of some of the peaks from Chrome Impute. Here's the one that someone asked about before. This is just looking at, now we're doing it across the entire genome, and we're looking at Chrome Impute versus Avocado on a chromosome by chromosome basis, and it's fairly consistent. I'm not going to go through that one, but we looked a little bit at the performance when you have more assays. Well, actually, maybe I'll just say the answer. So basically, what this shows is that as you get more and more assays for a given cell type, the difference between Chrome Impute and Avocado gets larger. So it does a better job of making use of lots of available data. It's sort of the converse of what I was saying before about Predicted, where we do care about being able to predict well when there's not very many data types available, but when you have lots of them, it can make use of them. And then finally, what we're looking at currently is some of the stuff like Yan-Jun was talking about, where we can try to make sense out of what the model is doing. And so the idea is that maybe these latent representations will be useful for lots of other techniques. So we're thinking of Avocado now not just as an imputation method, but as a general way to try to put all of the Encode and roadmap data into a sort of a computable representation that you can use for lots of different downstream tasks. So for example, if you just take the latent factors for the cell types and do a TS&E projection, they all break out very nicely by the tissue type. So it seems like the latent factors are capturing something about cell types. If you try to do an RNA-seq prediction task, so this is a task taken from the Roadmap Epigenomics paper, and you're just binary classification, kind of like what Yan-Jun was talking about. Here we're just using, I think it's gradient boosting as the predictor, but the input is either the roadmap data itself or the latent factors. And the latent factors give you much better predictive performance on this task. You can do a similar thing. Now we're looking at three-dimensional structure. So Bing Ren's lab described these. This is a heat map from a high-C experiment showing the contacts between pairs of DNA sequences in the genome. And a fire is a frequently interacting region, fire. And so Bing Ren's lab identified all of these fires in the human genome, and Jacob said, well, how well can we predict whether or not something has a fire score greater than 3. Again, this was a task that was taken from the Ren Lab paper, and we just asked, could we do a good job of that if instead of using the actual epigenetic data, so we have like 7 histone marks, and we do gradient boosting, if we take the Avocado imputed data, the Chrome imputed data, the actual epigenetic data, or the Avocado latent factors, we do a much better job of predicting where the fires are in that setting as well. And then I'll just say that we're now looking at using things like integrated gradients, which is one of these post-processing techniques for understanding what the model is doing. This is just a motivating example where the different colors for this method show you which pixels are helpful in discriminating between, saying whether or not this observation is a 9, for example. And so we can use this to take the imputed values, which are shown at the top here, and subdivide them according to the components of the model. And I've got some slides, but I'm not going to go through them, that sort of show that we can then tease out potentially interesting characteristics of, let's say, a particular peak. This looks like it has a lot of cell type specific component to this peak, or it does not, and so on. And so we're hoping that we can use that to come up with interesting biological hypotheses. So I'm going to stop there, and I'm happy to answer more questions.